Hey guys, Mr. McDonald here. We need to talk about two things this week. And the first video that we need to talk about gets into that first big topic of the week, and that's the physiology of neurons. Last week we talked a bunch about the different types of neurons. We got into nerve cells, and we talked about how the nerve cells have axons and dendrites, and the fact that they're one-way roads essentially, and that the electrical impulses can only move from the dendrites to the axons, which is why the arrows on your assignments were so important for you guys to understand that. All right, so that kind of brings us to our first slide today, and our first slide today talks about the functional properties of a neuron. And neurons basically have two jobs that they need to do. The first is irritability, so they have to be able to respond to stimuli. And the second one is conductivity, which is the ability to transmit an impulse. So they kind of go hand in hand. The whole irritability part means that something in the environment, some type of stimuli, has to actually create an electrical impulse. And then once that impulse has been created, we need to be able to conduct it somewhere, which is usually to the central nervous system. So these two properties of neurons are very much tied together, and are very much reliant upon the generation of electrical impulses within the neuron. So that's what we're going to get into today, how we generate those electrical impulses. Okay, so what we're going to start out with is what we call a resting neuron. And the plasma membrane of our neuron at rest is polarized. And if you think back to a water cell, polarized means that we have an end that's positive and an end that's negative. And as it turns out, we have fewer positive ions inside the cell than outside the cell. So what that means is it's slightly positive outside and slightly negative inside our cell membrane. So that gives us a polarized environment. And that's where our neurons are. They're not conducting an electrical impulse. Okay, so again, thinking back to what we learned last week, our electrical impulse comes into our nerve cell at the dendrites. The dendrites are going to detect the impulses coming from receptor organs, from other nerve cells. And what happens is when they get enough stimulation, again, enough electrical impulse, that builds up, and when those charges get high enough, we get to a tipping point. Now, we talked about tipping points with muscles, if you think back. And when we hit that tipping point, an electrical impulse was generated starting at the axon hillock, which was also something that we had to label and have in our diagrams of the neuron last week. So again, if you're not sure what those terms mean, go back and look at your diagrams from last week. Okay, so once we've reached that tipping point and an electrical impulse is big enough at our axon hillock, it gets into this resting axon, which is what you guys are seeing here. All right, so I don't know how well you guys can see, and I don't usually do this, but I'm just going to read what it says. It said the resting membrane is polarized. In its resting state, the external surface of the membrane is slightly positive. Its internal face is slightly negative. All right, so the extracellular ions that we use are sodium and potassium. And again, our membrane is impermeable to both of these ions. So that means we need proteins in order to move them in and out of the cell. All right, so depolarization happens. A stimulus has depolarized our neuron's membrane. Okay, so the membrane is now open to sodium as sodium channels open up. So let's go to a picture here. So here's what's happened to set this picture up. We've hit a tipping point in our axon hillock, and an electrical charge has now opened up little proteins in our cell membrane. And once it opens up those proteins, much like a mechanical garage door opener, the shape of those proteins allows sodium ions to rush into our polarized cell membrane. So that's what we're seeing here. And when that happens, the sodium ions are going to rush in, and they're going to cause our polarity to flip. All right, so that's what we see here. It's starting to flip. So what does that mean for us? All right, so the movement of ions, again, that's the sodium ions rushing in, all right, initiates our action potential, our electrical impulse. All right, and what that's going to do is it's going to open up those little, again, proteins that are embedded in our cell membrane, and we create what's called a graded potential, and that means that our membrane is now more positive inside than outside, so it's flipped. All right, and that's what you see here. Now, once this happens, the electrical impulse that we see here begins to move down our axon, and it keeps opening up more and more of those little proteins, and allows more and more sodium ions to rush in. Okay, and we call that propagation. All right, so that's what you're seeing here. And let me go back again. So if enough sodium, our impulse is strong enough, all right, gets into our nerve cell, we begin to propagate and move down the entire length of the axon. All right, these impulses travel very, very fast. They travel faster when there's a myelin sheath, but again, this comes from an electrical impulse reaching a tipping point, and all that's happening from a microbiological standpoint here is that we're opening up little ion channels, little proteins in the cell membrane, and allowing sodium ions to rush into our cell. And that's what we're seeing happen here. Now, it's an electrical impulse, so somehow we need to undo this depolarized state, and we call this repolarization. Okay, so at the same time that the sodium ions are rushing into our cell, potassium ions are rushing out of our neuron. Okay, so that's going to help us in repolarizing our membrane. They both have a positive charge. All right. Repolarization involves restoring the inside of the membrane to slightly negative and the outer surface to a slightly positive charge. One of the big players in this is a familiar name that we've seen before. Okay, and that's the sodium-potassium pump. All right, so we get back to our initial conditions, all right, using the sodium-potassium pump. So this requires energy. That's the second bullet point. So this pump using ATP restores the original configuration, as we learned about before, 3 sodium ions and 2 potassium ions at a time. All right, so again, this requires ATP, it requires energy. And this is how we generate an electrical impulse. And this is just a quick diagram of our sodium-potassium pump. And that removes all the sodiums that rushed into our cell, and it brings the potassiums back into our nerve cell. Okay, so that's what we see happening here in our sodium-potassium pump. All right, so what we've covered in this video is how we generate an action potential or an electrical impulse that travels the length of our neuron all the way to our axon terminal. We'll get to the axon terminal in our next video. Okay, so if you have any questions, please email me, let me know. And again, this is one of the big ideas of our chapters, generating an electrical impulse in our neurons.